Hi class, welcome to the Chapter 3 lecture. The topic of Chapter 3 is on compartmentation, so basically membranes and the movement of material through membranes. We are really going to focus on the three sections highlighted here in the pink font. So we'll spend some time reviewing the structure of a membrane, and then we'll talk about the different compartments and apply that to some of the tissues of the body. So we're actually going to get into our first organ system tonight as well. We will learn about the integumentary system, which includes the skin. So most of the content really focuses on pages 61 to 87 in the 8th edition book. But again, those three center sections of the textbook. So as you know, the cell membrane has two very important jobs. One job is to separate the inside from the outside, right? It defines that barrier from the internal world versus environment. The second job of the cellular membrane is to regulate the passage of materials that go through it. So two important functions of every cell membrane. It creates that internal versus external environment and then regulates the movement of certain materials through it. Additionally, there is physical protection provided and the creation of these compartments. The cell membrane is important in helping to regulate the movement of materials across it. So that ties into the communication of one cell to another cell as well. And then additionally, the cell membrane does provide some structural support for the cell. Not as much as we see, say, with a cell wall in a plant. That would be very rigid. But the cytoskeleton does provide the appropriate amount of support for the cell. And also some cells are involved in the secretion of materials. And secretion is when movement of anything happens towards the out of the body, so from the cell outward. So the cell membrane has been described as the fluid mosaic model, which is appropriate. Let's take a look. So looking at the structural integrity, we are first of all reminded that the cell membrane exists as a bilayer, right? There's two layers that are inverse to each other. It is composed of phospholipids, which have the glycerol head and then the fatty acid tails. So we have very defined regions where there is, where the cell membrane interacts well with water in those hydrophilic regions on both sides of the membrane. But the internal characteristics are hydrophobic. So this is the fat soluble portion of the membrane. You can see sandwiched in, this is cholesterol. So that's where cholesterol exists in its normal, natural, healthy place, is right there in the cell membrane. And cholesterol is important because it is used to make the variety of steroids that our body needs. So now let's take a little bit more of a look into the other materials that are present in the cell membrane. So you can see that there are a number of different, these are different proteins. This is a transmembrane protein because it goes across the whole membrane, right? So there's regions in the external environment as well as in the internal environment. In this case, this transmembrane protein is anchored to the cytoskeleton, which means it has a role in providing structural support to the cell. There are other proteins that are present only in certain portions of the cell membrane. So we have these lipid anchored proteins that span to the inside and outside of the membrane, but they are anchored within that hydrophobic region of the cell. Still yet on the outside, protruding to the external environment of the cell, we have a variety of glycoproteins. And just like the name suggests, you know what a protein is. Well, glyco means sugar. So these are proteins that have sugar groups attached to them. And we'll see what some of the functions of those are. But so you can begin to appreciate that there are a number of different proteins that are present, embedded within the cell membrane. If we look over to the right side, we're looking at something that looks very busy. This is a transmembrane protein, and it goes across 7 times. So there's a whole class of proteins that fall into this group that transverse. So if we begin at one end of the molecule and count how many times it goes back and forth across the membrane, it goes across 7 times. Now anchored within the cell to this protein, we see phosphate. And anytime you see a phosphate molecule, I hope that you're thinking of ATP, of energy, since phosphate is part of that. So this is looking at that 7-membrane protein a little bit closer. So every protein has a carboxyl terminus and then an amino terminus, and then it goes across 7 times. Right, so there's regions of this protein that are interacting with the hydrophobic regions of the membrane, as well as with the hydrophilic regions of the membrane. So this is a major class of proteins that is found in the cell membrane. All right, so let's look at the cell membrane a little bit differently. So now we're taking the cell membrane and really breaking it up by the components of which it's made. So there's cholesterol. We have the phospholipids. There's another group of lipids. We have carbohydrates and then proteins. So the combination of a carbohydrate and a protein forms what is called a glycoprotein. The combination of a carbohydrate with a lipid makes a glycolipid. So we have a number of different proteins that are present in our cell membrane that allow for the movement of the various materials that need to go past or need to go through the membrane. So both glycolipids and glycoproteins, they are involved in providing structural support. So that means they're anchored to the cytoskeleton. They are involved in cell recognition, so the interaction of one cell to another cell. And then the immune system utilizes a number of glycoproteins and glycolipids in the activation, as well as the specific immune mechanisms that protect us. Moving over, the lipid bilayer would include the phospholipids as well as cholesterol, right? We saw that in this figure. So we've got our phospholipids and then the cholesterol. That's where cholesterol naturally exists. And this is important in creating that lipid bilayer, which is really important. So let's just take a minute and think about the cell membrane. If everything were hydrophilic, water loving, there would be no such thing as a cell, right? The majority of our body is water. So if that cell membrane were only hydrophilic, water loving, it would dissolve within the watery environment of our body. If it were only hydrophobic, then material would have a very hard time moving through it. We'll review the characteristics of the different materials that move through the membrane. All right, so your book uses a number of these like flow diagrams or concept maps that allow you to see the components that are used to build a certain structure, but then we'll also see different concept maps that relate to the function as well. All right, so within our cells, we have compartments. And then within our body, we have other compartments as well. And this is necessary because as we grow, right, our body changes, our cells changes. And that's what the process of differentiation is. And it just simply means that certain genes are turned on at a certain time, and that leads to the creation of a certain cell. So we can begin with a stem cell that can produce any cell, any blood cell. But through the process of differentiation, that cell it becomes committed to being a certain type of cell. All right, so if we look at the different compartments of cells, of course, the cell membrane is one of those. And then we would have the cytoplasm, which would include the cytosol, as well as inclusion bodies. We'll get to what those are. And then the cytoskeleton and the various proteins that are utilized to help support that cytoskeleton. So cells are divided into major components. We've got the membrane, the cytoplasm, and then within the cytoplasm, we've got organelles, and that really depends on what type of cell we're talking about. And then, of course, the major third component is the nucleus. So we have the cell membrane, the cytoplasm, and then the nucleus as our major components or compartments for eukaryotic cells, for human cells. So looking at that in a version of a concept map, we have the nucleus, the cytoplasm that is external to the nuclear membrane, and then the cell membrane surrounds it all. So, right, our cytosol is just the fluid portion of the cytoplasm. So you can think of the cytosol as equal to the cytoplasm minus the organelles that are present in it. It's just the fluid. We have a number of different organelles that have very specific functions within our cells, right? We've got the mitochondria, the reticulum, the Golgi apparatus, lysosomes, peroxisomes. Inclusion bodies would include different lipids and as well as glycogen, and also ribosomes fall into this category. And then examples of the protein fibers would include the cytoskeleton. During cell division, the cytoskeleton grows from the centrioles to provide the tracts so that our cells can divide. And then also cilia and flagella are examples of different protein fibers. So that's a way to conceptually organize, to map out the different components of the eukaryotic cell. We have our three major compartments of the nucleus, the cytoplasm, and then the cell membrane. All right, so what exactly is an inclusion body? Well, they have no membranes, so they're not an organelle. They can be used to store different nutrients. So we have inclusions that store glycogen, which is one of our energy reserves. So those are called glycogen granules. And then we have other inclusions that store different lipid materials. There are others that have functions independent of nutrients. And then we get to our ribosomes. So ribosomes are an example of an inclusion body. Again, that means they have no membrane. Ribosomes are made of protein as well as the RNA that's running through it. They're involved in, of course, producing proteins. Proteins are made at the site of a ribosome. Without a ribosome, we cannot produce proteins. Some of our ribosomes are free in the cytoplasm, whereas others are attached. And then we've got polyribosomes also, right? Poly means many. All right, so let's kind of run through quickly the eukaryotic organelles that are important in physiology. So hopefully some of this is familiar to you. So those that have membranes, of course, include the nucleus, the mitochondria, the Golgi, the rough ER, as well as the smooth ER. And then we get to lysosomes, which have no membranes. right? And then we get to our inclusion bodies. Lysosomes have a very important job in helping to break down materials in our cells. So examples of the non-membranous organelles, the ribosome is that classic example, but so are elements of the cytoskeleton. And we find those in the cilia, the flagella, and even in the microvilli of cells that have the function of absorption, like in our intestine. So let's look at that. So we're looking at quite a large cell here, right? It's shaped like a rectangle. We can see the villi and the across the top of it. And all this is a way to increase the surface area to volume ratio. So we see that that's actually one of the major themes in biology and physiology, is this concept of cell surface area divided by the volume. So the surface area to volume ratio. And many different cells in our body maximize the surface area to volume ratio by having wiggles in them like we have here. So we have the rippling of the villi, and then even those ripple out into the microvilli. But this is seen over and over again in biology, not just in our species. Oopsie, let me get my mouse back. There we go. Maybe not. Just a second. Tends to get lost. Oh goodness. Okay. There we go. All right. So looking over on the right side, we've got our villi, and each of these villi actually have a blood supply into them because they are, the function is absorption, the movement of materials across them. We can see examples of the cytoskeleton. We've got a mitochondria here, and we have different components of the cytoskeleton as represented by the different colors that are here. We can see the nucleus and endoplasmic reticulum. Our Golgi apparatus is this structure. We've got free ribosomes, and then there are also ribosomes on the rough ER, as well as on the nuclear membrane itself. So looking at some of these different structures, you've got a nice table or graphic here that shows you what they look like and where they're found, and then reviews the different characteristics as well as the function of each of these components of the eukaryotic cell. So I'm not going to read through this here, but hopefully this is stuff that's sounding a little bit familiar to you from your introductory biology course. But please take a look. You've got figures in your book. These are from an earlier version of the textbook, but they're essentially the same as what is in the current textbook. All right. And then it continues on, right? We've got more different subcellular structures and their corresponding characteristics and functions. All right, so now let's remind ourselves about the mitochondria. And the mitochondria is all about energy. And when we talk about energy, we need to think about ATP. The mitochondria has two membranes. So it's got this outer membrane, and then it's got an inner membrane. And what that does, very important, is it creates a compartment within the mitochondria. So we end up with this intermembrane space, right? Here's the inner membrane. These are called cristae. It's the inner membrane, all wiggly. And then the outer membrane is smoother. But that inner membrane that has all those folds, it's all about increasing the surface area to volume ratio. Why do we want to do that though? Why is that important? Why is this a concept that is conserved and seen over and over again in evolution? Well, the simple answer is because it works, right? We wouldn't see something selected over thousands of years, millions of years of evolution if it was a defective process. The machinery that does cell respiration, it's all located within this inner membrane. So by having more inner membrane, that means there is more cellular machinery to do the job of the mitochondria, which is to process energy. So that's what it gets down to. We have more material to do the job. So it makes this organelle, the mitochondria, more effective in the production of energy. Let's look at how a tissue is formed, right? We've been talking really about, well, organelles or a cell, but tissues are made of of cells. Sometimes they're made out of the same type of cell. Other times they have different types of cells within them. But each of the cells that form a tissue has different connections. And we have different connections. Again, that means that they have different functions, right? We wouldn't have different types of connections if they weren't needed. So one of those is called a gap junction. And gap junctions are like tunnels between the cell. I'll show you a picture in a moment, but these are little tunnel passageways. So fluid and whatever's dissolved in that fluid can move from cell to cell through a gap junction. There are also what are called tight junctions. I think of these like a zipper, right? Zip it up tight. These prevent the leakage of material, specifically water. So they help to bind together two cells tightly so that there is not loss of material from one cell to another or to the environment. So there are a couple different types of tight junctions. We have what are called occluding junctions and anchoring junctions, but they both prevent the free movement of water. And then we have another type of junction called a desmosome. And these are made up of a protein called cadherin. And these are found in certain tissues more than others. So we see these in the heart muscle specifically, but they're also found in our skin and other organs. The characteristic of the desmosome is it's a very tight junction to adhere two cells to each other, but it stretches. Now let's apply that to a biological clinical example. Think about your heart. Every time it's pumping, what's happening? Boom, boom, ba-boom, ba-boom, ba-boom. Yeah, that heart muscle is stretching. So having the desmosome there, it allows for that maximum stretch so that we can get the function out of it, but without it tearing. So three main types of cell junctions: the gap junction, the tight junction, and then the desmosome. So we can look at, let me point them out here. This flow chart shows a little bit more than what we were talking about. So gap junction is right here under type. So gap junction, next door is tight junction, and then we have adherens, we have the desmosome. So we'll begin with the type, since that's what we just learned about. So if we want to kind of back it out to a broader layer though, each of these types of junctions serves a function, right? So we have some that are involved in the communication of one cell to another. That is the gap junction. It is a tunnel. It's a, oh my gosh, I was just gonna say it's a hose, and that reminded me I turned on my hose and left it on an hour ago. So excuse me for a moment. I'm gonna pause the record and go turn off my hose. All right, well, sorry about that brief pause. That's one of the advantages of teaching from home, is I could just run out and turn my hose off. However, if we were on campus at San Elijo or main campus, that would not be possible. Anyhow, so a communicating cell junctions that are used for communication are correctly represented like a hose running from one cell to another, and they utilize the gap junction. Occluding junctions, well, what does occlusion mean? Occlusion means blocking a pathway. So that's opposite to what we saw with the gap junction. So the tight junction is what prevents the seepage of water and whatever's dissolved in that water outside of the cells. And an example of that is with the protein actin that we'll study and learn about when we get into our musculoskeletal system. And then the desmosome was the third type of cell junction we studied, and it is an example of one of the types of anchoring junctions. And again, I'll use the example of the desmosomes that are found in the heart and in other tissue that stretches. Their job is to allow that stretch, but then importantly, it returns to its pre-stretch stage. So a desmosome, which is found in the cardiac tissue, is a very good example of that. So as you can see, there are other types of gap junctions, and they have their different functions, and there certainly is some overlap between them, but we're going to focus on the gap, the tight, and then desmosomes. So now let's look at the picture. It's kind of hard to conceptualize. This is the example of the gap junction, which is like a hose or a tunnel or a pathway that connects one cell, cell 1, to cell 2. So you can see that there's a direct connection. It's like there's a straw or a hose connecting these two cells. So material can move back and forth. In panel C here in the middle, we're looking at an example of the tight junction, the occluding junction. And this figure actually kind of annoys me a little bit, honestly. And the reason why is it makes it look like these are the tight junctions here and here. But the way the figure kind of makes it look like is that there are gaps, and really there aren't. The thing about the tight junction is it's like a reinforced stitching. Like, look at your jeans. There's not just one low line of stitching in them. There's two, and they're reinforced. So tight junctions are there to prevent the seepage of fluid. And now look at the anchoring junctions, a bit more complicated now. Well, these anchoring junctions, such as the desmosome, they are attached to different components of the cytoskeleton and different glycoproteins. So that allows them to have their function of maintaining the integrity of the junction and cell through stretch. All right, so now we're going to switch into our first organ system, which is the integumentary system, skin, hair, and nails. It fits really well in this topic because of the different types of junctions that are present in skin. So we have 4 different types of tissue. The epithelial tissue, we've got connective tissue, so blood and bone are examples of that. We have muscle, and then we also have nervous or neural tissue. So there are 4 tissue types, major tissue types in humans: epithelial, connective, muscle, and nervous. And they all have different functions. So our epithelial surfaces, well, where are they found first of all? They are on all of our external surfaces as well as our internal membranes. So epithelial tissues are commonly called epithelia. And they have such an important job, right? They ultimately are what separate us from the outside. Our skin does that. The structure of the epithelial, it depends upon where in the body we're looking, but we have different cell shapes of the epithelial as well as different arrangements of them. Epithelial, such as our skin, the dermis, epidermis, dermis, and endodermis, they are separated from other parts of our body by what is called a basement membrane. And the way I like to think about basement membranes are these are really good separations. Material that bypasses a basement membrane has to do it by specific mechanisms. We'll really come to understand that when we move into the respiratory and renal sections of our course. So some of our epithelial is tight, meaning it does not allow for the leakage of material. On the other hand, we have other epithelial that is leaky or may be induced to be leaky so that it can serve a function at that time. All right, so let's look at how epithelial cells are classified. So we're talking about those external and internal surfaces that shield all of our body, internally and externally. So this would include like our intestine. It has a hole in it, right? So we've got our internal epithelial that lines that passageway. Some of the epithelial cells secrete material, and some don't. It depends upon what cell we're looking at. All right, so how can we describe the epithelial? So if we look at the histology, which would involve looking at different cells under the microscope, and that's what we would be doing in lab this week, part of our lab, we can classify it based on layers, and we've got two options. Simple means there's just one layer. Stratified means two or more. So we can classify it based on how many layers it has. And then we can also look at the morphology or the shape of the cell. And we've got three options there: squamous, cuboidal, and columnar. And pretty much every time we see columnar epithelial cells, it's serving the same function. When we see cuboidal cells, serving the same function. Same with squamous. Let's take a look at what they look at. Okay, so this is a simplified kind of cartoon representation of the human epithelial systems. So we have our skin, which is the external barrier, right? I think we can all conceptualize that the skin is that outer barrier. But inside we have all of the various organs and passageways, tubes, and cells where material moves through. So our respiratory system is open to the external environment, right? We breathe in oxygen, we exhale carbon dioxide, but the lungs exchange that material into the bloodstream, and then those materials, oxygen and carbon dioxide, move back and forth between the cells so that we can supply our cells with oxygen and move away the products of respiration, which would be carbon dioxide. We see also there is exchange at the level of the kidneys. This is actually one of the major exchange sites in our body. Our kidneys are really important in recovering materials that otherwise would be lost in our urine. But instead our kidneys bring it back in. So we do have movement of material from the kidneys to and from the bloodstream. Our digestive system, of course, we eat or drink, and that's the pathway from the external environment into the internal components of the digestive system. So that would include all of the stomach, the large intestine, the small intestine, and all of the accessory organs. There is exchange between the blood and the digestive system. And that's absolutely necessary. I mean, what would the point of eating be if there were no way to get those materials from what we eat and digest into our body? So yes, the digestive system is a major source of exchange too. So all over our body, we've got interaction and exchange of material between it and the environment. Now, looking at these, the key down here, secretion means the movement of material out of the body. So we're secreting it into the bloodstream. And then exchange is represented by the double black arrows here. So the intestine, the urinary system, the respiratory system, and then the reproductive system as well. Now let's look at a picture of the skin. So we can see there's a lot going on. Let's divide the skin into its respective layers. So the top part is the epidermis, and it consists of mostly dead cells and proteins that make it waterproof and durable. The skin So the epidermis, let's look at what's in here. There aren't many blood vessels. There are no nerves. And that's good. I mean, we would all be, we would dehydrate. We would all have significant pain if those structures were present in the epidermis. Once we move below the epidermis into the dermis, right, this is the biggest section of the skin, now we see blood vessels. There are blood vessels. We have hair follicles. There are glands that secrete material through pathways to the skin. So we've got, this is where the blood vessels and the nerve endings are. They are protected below the epidermis. The epidermis protects the important structures of the blood vessels and the nerves, which are present in the dermis. Below the dermis is the hypodermis, right? We've seen this prefix before. Hypo means less than, or in this case, below. The hypodermis is where a lot of the connective tissue is. So we've got adipose tissue. This is healthy fat. It's brown fat, which provides energy reserves. And also we do have some of the glands that produce the different secretions. So for example, each hair follicle has a gland that produces oil so that the follicle has the ability to move out without causing friction. So here's a good picture of that. So we're looking at a hair follicle here, and then it's got the ability, keratin is one of the proteins in both our hair and our skin that allows it to be waterproof. But what would be the point of producing a protein if there wasn't a secretion mechanism to get it where it's needed? We're looking here, just another structure I'd like to point out. This is called a sebaceous gland, and these are just glands that release lipidy materials. And then finally, I'd be remiss not to specifically point this out. In the dermis, we have a number of sensory receptors, which are critical in the healthy functioning of the human body. For example, what would happen if I took my finger and put it on a hot stove, right? Say this is the stove and it's super hot and I touch it for whatever dumb reason. What's going to happen? I'm not going to just leave my finger on that hot surface. Why not? Well, the sensory receptors that are present in my skin are going to perceive that pain and send a signal to the muscles in my arm to move my arm away. So that's a very important function of the epithelial tissue, specifically the skin, is that this is where a number of our sensory receptors are, which are critical to allow us to safely interact with our environment. So looking at a little bit simpler of a picture, we've got the epidermis at the top, and these are primarily dead cells, dead skin cells that have keratin, that protein that gives the waterproofing ability. Below that, see, we've got different junctions here. See all the junctions between the cells. Every cell is connected. So we're looking at desmosomes in the skin, because the skin is one of the places where we see desmosomes as well. And those very tightly anchor one cell to another, but they allow the stretch that is necessary in the skin, right? We've all had examples where we have had swelling of, say, an ankle or a wrist or something, right? Imagine what would happen if your skin did not stretch and you have swelling from inflammation. If your skin doesn't stretch and allow that, it would cause something called compartment syndrome, which is a true medical emergency. In compartment syndrome, the internal structures are being crushed because of excess pressure on them. So the way to relieve it is to cut open the skin, cut open it so that pressure is relieved. All right, also down towards moving through the epidermis to the dermis, down to the hypodermis. The hypodermis is where we see some of the other structures. We have our basement membrane down here. So this thicker kind of blue line is the basement membrane. And every epithelial tissue has a basement membrane below it. So think of that like a tight barrier. Oh, I just saw one of my kitties go up the stairs. Sorry, I got distracted. This is an example of a melanocyte, which is the skin cell that produces melanin, which gives us the coloration of our skin. So looking at those junctions, these desmosomes, or specifically the hemodesmosomes, they are attaching one cell to another, but they also anchor the epithelial into the basement membrane. And that is a very important barrier. All right, so just a couple terms that I want to differentiate for you. You've probably heard of these. So necrosis, excuse me, is simply rotting. Sorry, but it's kind of gross, but that's the way I like to describe it. If our cells just died whenever they did, it would lead to necrosis. And necrosis is something we never want to have in our body. Necrosis will lead to infection. It can lead to blood clots. It can lead to all kinds of problems. And what happens is the lysosomes, which are one of those structures inside our cells, they're involved in the digestion of material by containing digestive enzymes. But what happens in necrosis is they just go crazy and everything becomes digested, including the healthy tissue. Apoptosis is completely opposite. It is a healthy process. In fact, apoptosis is a function of our healthy immune response. It is programmed cell death. It is cell suicide, which sounds drastic. But what it really means is that every cell has a time in which it is programmed to die to prevent necrosis. So our red cells live for, on average, 120 days. At 120 days, they undergo apoptosis so that they are removed from the body in a healthy way so that they don't just fall apart and degrade and cause rotting or necrosis. So apoptosis is healthy, necrosis is not. Necrosis is pathology. Apoptosis or apoptosis is physiology. That's healthy. I've heard the word pronounced equally as apoptosis and apoptosis. It seems to just be a preference by whatever immunologist you're talking to. All right, we're almost done. So a little bit about stem cells. So the true stem cell can give rise to all of the blood cells in our body, right? It comes from the marrow and it's very immature and it can differentiate into anything, any of the blood cells based upon the correct conditions. That is differentiation. So not all stem cells are created equal. We have some that are way younger, and that means they are capable of going to many, many different pathways. So genetic differentiation is the process of regulating protein expression in a cell. It allows the bone cell to be bone and the skin cell to be skin and the intestinal cell to be the epithelial cell that makes that up. So genetic differentiation is also part of our aging process, right? We don't look like big babies, right? We don't all look like big chubby cheeks and the big giant, well, I'm just gonna shut up about that part. I was gonna say we don't all have that big giant belly, but as adults, well, I find myself having one. But we look very different from how we did it as babies. Right? And then we look different in the infant stage versus the child stage versus the adult. That is another example of differentiation. Different cells are active at different times in our life, and that changes our phenotype. It changes our physical appearance, which is what phenotype is. So I'm going to leave this for you. We will revisit this throughout the semester. But again, not every stem cell is created equal. So I'd like you to look up what is a pluripotent stem cell versus a multipotent stem cell and what the differences are. And we'll talk about this in a future activity but I'll leave that with you to dig into for now. All right, that actually wraps us up. That's it on chapter 3 on compartmentation. We will pick up a little bit more with this topic in chapter 5. So we're not quite done learning about compartmentation, but hopefully that was helpful. I hope you have a great day and stay safe. Take care, guys. I'll see you soon. Bye.